Welcome to our video on experimenting with cellular respiration. Today we're doing a lab which involves recording the rate of respiration on mung beans. So before we start the lab, make sure you put the thermometer in the water bath just so that we know if the temperature changes.
First, we need to prepare the equipment used in this lab. In this lab, we are using respirometers. Grab a pipette and wrap parafilm around 1 inch from the blunt side of it. You can stretch the parafilm to get a tighter adhesion onto the pipette. Once done, make sure to apply Vaseline to the blunt end of the pipette and insert it into the string.
Before using, we need to check if the seal is tight and will not let any water through. Place the tube onto the stopper and put the entire setup fully underwater for at least 20 seconds. If bubbles are forming, that means there is water leaking into the tube. If that happens, check the seal between the pipette and the stopper and tighten the seal.
Next in testing, place the absorbent cotton in the tube, making sure to push it all the way to the bottom. Then, soak the cotton with 1 drop of a KOH solution as shown. After putting a layer of KOH in, make sure that you put a layer of non-absorbent cotton.
Next, we must place our variables inside the tube. These variables will vary per lab. Our variable will be mung beans. To not have any sources of error, we need to have the same volume of dry and germinated mung beans. This will be done through water displacement. Place around 30 milliliters of distilled water into a graduated cylinder. Next, place a certain number of soaked mung beans inside it. We will use 30. Once done, record the displacement of the water and place the mung beans inside one of the 3 respirometers. Measure out the same number of mung beans as the soaked. Since the displacement isn't reached, we use glass beads to compensate.
Lastly, do the same thing with the glass beads.
Next, we will need to equilibrate all the respirometers. Place all 3 of them in water, making sure to leave the tips out.
Equilibrating for 5 minutes, add 1 drop of the dye to the end of the pipette.
10 minutes. Okay, now with me.
We can conclude from our results that the soaked mung beans had the highest rate of cellular respiration and consumed the most oxygen compared to the dried mung beans and the beans. In this lab, our alternative hypothesis was that we thought the soaked beans would have a higher rate of cellular respiration and would consume the most oxygen. This was proven to be true from our results. A source of error we encountered in this lab is that we accidentally touched the respirometer after it had been equilibrated in the water bath. This factor could have altered the results given. If done again, we would correct our errors to ensure accuracy.